Good afternoon. Anybody have any questions from last time? Does anyone remember what we talked about last time? What was it? Antivirals, no questions? Okay. Come in. Today we're going to talk about one of my favorite topics, evolution. And by the end of this, even if you don't believe in it, you will. We're going to talk about where virus... Yes? Oh, antivirals? Sure. Neuraminidase. Yes.
When it removes the sialic acid when it exits
right.
the cell, does it prevent from removing virions to attach, or does it remove them permanently?
Okay. Got it. So the question is, the neuraminidase helps release the virion from the cell. Would it also prevent virions from attaching? And if it were in the same part of the cell, it would. But remember, that cell is infected. It's producing virions. There's no need to be reinfected, most likely. The sialic acids will regenerate in time, but the cell that's producing virions doesn't have to be reinfected. In fact, many viruses go to great lengths to prevent reinfection of the same cell. It's a waste of genetic material. So that's not an issue. Okay, today we're going to talk about where viruses came from. I don't know, but we'll talk about the evidence. Where they're going, don't know either, but we'll talk about hypotheses. How are they getting there, we can tell you. And how would we know? We can tell you that as well. So virology makes evolution very clear. So this is what I think most people don't understand, that if you can look at viruses in terms of their evolution, you will understand the rest of evolution, because this virology evolution happens in minutes. Whereas for everything else, it takes a long time. And you'll see why that is today. So virus populations, well, host populations grow and adapt. At the same time, there are virus populations that infect them, and they undergo selections as well. Every day, a new virus population emerges. You don't see this, of course, because you don't see a new disease or a new virulence or some other expression of a new virus, but they're emerging every day. Viruses can also be forces in the evolution of host populations. This has happened over time throughout history, and it's happening to this day. If a population can't adapt to a new virus that has arisen, it will be exterminated. So both the virus and the host have to evolve, and that's part of evolution. So the public always sees virus evolution, but they don't think of it as evolution. Right? The people who don't believe in evolution are seeing it before their very eyes. We have new viral diseases emerging. We have AIDS, we have West Nile virus, hepatitis C, Ebola. Flu every year comes back. Common colds, drug resistance of the sort we talk about. This is all evolution, right in front of everyone's eyes. And the fact is that viruses evolve faster than people can really comprehend. And even you who are in the know now, having taken a virology course, maybe until now you won't understand how this can be so. They evolve incredibly quickly. And so you can see evolution happening in a day. So here's an example. I gave a lecture at Lehman College a couple of years ago. And there was a professor in the audience who, I'm not sure what he did, but he knew a lot about evolution. One of the projects he worked on, he went to the Bronx River and he pulled out life. And his lab was full of jars of fish and lizards and, not lizards, frogs and all kinds of things. And he was comparing how the life evolved in the river compared to, say, 40 years ago. So I showed an experiment which showed how viruses can be selected very quickly for drug resistance. And I said, this is evolution in a test tube in 20 minutes. And he said, it's not evolution, it's selection. So even a guy who has studied evolution of animals his whole career doesn't understand that selection is a huge force. And even if it happens in 20 minutes, it's still evolution. So if he can't figure it out, this guy's a PhD, he's a professor, how can the public figure it out? So viruses will really clarify all this, and that's why you've taken this whole course now to get to this point where you can understand everything I'm going to tell you and understand how evolution proceeds so quickly. So another problem with the public is viruses are tiny, they're passive, and people will say, how could they produce these responses upon infection? How could they evolve? It's just too small, they're just molecules. Well, the diversity of genomes that are produced in a cell is just something people can't comprehend. Beyond the fact that they're tiny and passive, how could they generate such diversity? It's not possible. And in fact, we are just beginning to understand the forces that drive evolution, scientists that is, why old and new viruses emerge, how viruses participate in the ecosystem. You know, the ocean is full of viruses, as we said the first day, and it's a huge player in the global geothermal cycle. It releases tons of carbon every day, and that's something we only learned in the last few years. And of course, what this all means for our own survival. Evolution of viruses is a relatively new field because it involves nucleotide sequences which we haven't been able to study for more than the last 30 years, let's say. So what is viral evolution? It's the change of a viral population in the face of selection pressures. So you see, Lehman College professor, evolution is a matter of selection. If the population cannot change or adapt, it disappears. And that's for the virus as well as the host. Virus populations do change. I'm going to say this about 20 times today, but it's very important that you remember that. And they display spectacular diversity. I mean, really spectacular. The numbers like you've never seen before. One guy in Spain who studies, one professor in Spain who studies virus evolution told me once, people seem to think that virus evolution is the same as the evolution of a cow or a fruit fly. But it's not. It's completely different because they have such spectacular diversity. And what is the source of that? That's something we're going to talk about today. Mutation and recombination. And viruses are the kings of doing this, much better than anything else on the planet. So, virus evolution then is defined in terms of a population, not an individual particle. We don't look at individual species of animals and define evolution. We look at the whole population, and you'll see why today. The populations are mixtures of mutants of all kinds that are made in huge amounts. So don't even think of an average. Don't look at a virus as an influenza virus and think of an average as a representative of the population. There is no average. There is no single particle that looks like the average. And virologists in the end are population biologists. Even though we think we are studying individual viruses, we are actually studying the whole population, how it interacts, and how it affects hosts. When it comes to viral evolution, the median is not the message. I don't know if you guys know that quote, right? Marshall McLuhan. When I was in college, this was a famous quotation, the medium is the message. You remember? Yes. Marshall McLuhan was a media guy. He said, the medium is what's important, not what's in it. TV, radio, newspaper. So Stephen Jay Gould, who is an evolutionary biologist, changed it. And it's brilliant. The median is not the message. So whatever is the average in a virus population isn't what's important. It's the whole population. Every individual is a potential winner in a virus population. Even the one with one mutation that may be obscure could end up being the most common after a single selective event. So the average is irrelevant. So today we're going to talk about 4 of the main drivers of evolution. We're going to talk about large numbers of progeny, which we've talked about already, but we'll codify it in terms of evolution. We're going to talk about mutants, a lot of mutants, and you will understand how these arise because we've gone through the molecular mechanisms of them. We're going to talk about the quasi-species and what that affects. And finally, we'll talk about how a selection is applied to change populations, to change virus populations. And if you've ever been confused about evolution, after today, just think instead of virus, you could put your favorite organism in there. The concepts are the same, although the numbers are very, very different. So let's look at numbers of progeny. I've already told you that HIV infection in a day of an individual, you get over 10 to the 9th particles a day. So there's HIV production in the blood. This is the half-life of the virus, the daily turnover. Huge turnover. 90% of the virus turns over and a lot. And for comparison, hepatitis B, which is also prodigious, 10 to the 11th particles, even more than HIV, a short half-life and high turnover as well. So this is what we mean by the production of large numbers of progeny. And these are two examples, but just about every acute virus infection has the same pattern. These happen to be chronic infections that look a lot like acute infections. But poliovirus, for example, an infected cell can produce tens of thousands of particles. So it's characteristic of the infection that large numbers of progeny are produced. And when you make a lot of viruses and the host is trying to intervene, that's where you get selection and eventually evolution. So evolution only happens when you have mutations. End of the story. There's no way around it. If you don't have mutation, you don't have evolution. And these are produced, mutations are produced when you copy a nucleic acid. So we evolve as a species because we make mistakes. It takes a lot longer because we have a long generation time. We don't reproduce all that much. And we're DNA based. So it takes tens of thousands of years for us to evolve. But a virus can do it much faster, as I said, can do it in a day. So RNA viruses are the masters of mutation. You know this by now. We've talked about it a few times. All polymerases make mistakes, but those of the RNA viruses can't make corrections to the mistakes. So the mistakes stay, and that's where the variability arises. So the average error frequency for an RNA virus, 1 in 10 to the 4th or 10 to the 5th nucleotides polymerized. All right, so in some viruses that have a genome length of 10,000 bases, so if you had a frequency of 1 in 10 to the 4th, that would be 1 mutation per genome. Every time a genome was produced, it would have a mutation in it. And the mutations, in theory, are random, so every genome could be different. As we said last time, every 10,000 genomes, you have viruses with every possible mutation, in theory. So this led to what's called the quasi-species concept. And that is on the basis of a study that was done with an RNA bacteriophage called Q beta. So a virus that infects bacteria, very easy to grow, rapid generation time, high numbers of progeny. In 1978, a paper was published which said in part a Q beta phage population is in a dynamic equilibrium with viral mutants arising at a high rate on the one hand and being strongly selected against on the other. The genome of Q beta cannot be described as a defined, unique structure, but rather as a weighted average of a large number of different individual sequences. So I remember reading this paper in 1978. Where was I? I was just finishing my PhD. I had no idea what it meant. No one had ever seen anything like this before. In this paper, they provided evidence that the virus populations were quite varied, but I really didn't know what it means. And neither did most other virologists, fortunately. It wasn't appreciated, far ahead of its time. But now it has led to this quasi-species concept. Viruses are not species as we are that are very defined sequences. They're mixtures. Virus populations are dynamic distributions of non-identical but related replicons. All right? So dynamic because they're always changing. Every time they replicate, they change. They're related because Q beta is always Q beta. So there's a structure that the virus evolves within. There's a sequence space, as we call it. So Q beta doesn't become poliovirus. It doesn't become a DNA virus. It changes within a space, so they're all related, but they're not identical. And that's what a quasi-species is. And you'll hear it being misused often, but it arose to talk about RNA viruses. It also applies to a certain extent to DNA viruses because they also change, but far less than do RNA viruses. So let's picture this. That's the easiest way to look at it. Let's pull a bit. We have a tube of virus in the freezer. Let's pull a little bit out, and if we could sequence every individual RNA in that mixture, this would be what some of them look like. This is just a subset, maybe a 50 or so here. And this is the genome represented by a line, and single base differences are shown by different symbols. So every genome is different. Now, we don't have a sequence method that can pick this up in a population yet. So you can't really sequence individual RNA molecules. I think deep sequencing is getting close, but there is an error rate associated with deep sequencing itself, and it's not yet clear whether that exceeds the variation in the quasi-species. But the point is that every genome is different. And if you did a consensus, you would get a line, and no virus would look like the consensus, because every virus is different. So this line is an average of all the other sequences, and at many positions they're so infrequent that they don't show up in the consensus. So the consensus looks like a line with no changes in it. So that's what a quasi-species looks like if we could draw it on a paper. And of course, it's much more extensive because there are many, many more virions than this in a population. So what does this mean? What does a quasi-species do, or what kinds of implications does it have? Well, it means that when you do an infection in the lab with virus, or when you get infected, you're not getting infected with one virus. You're getting infected with a population of viruses. Right? All different sequences, all the time, no exception. And that large number is produced by the virus growing in a cell. So if I grow a virus in a cell in the laboratory, while it's growing in the cell, you have all these different RNAs made, and that's what comes out. When you get infected with influenza virus, you produce a quasi-species, and you infect someone else with the quasi-species. At every step, there is selection going on to change the population. So the virus, the quasi-species that you're producing is different from the quasi-species of the unfortunate person that you infect with the same virus. It's always different. And so the nature of that quasi-species represents the selection going on in particular cells and hosts. So it would be very interesting to be able to take an isolate from a person or a cell and sequence every individual genome, or maybe sequence 100 individual genomes, and see how they differ. And see how they differ from host to host, from animal to animal. We can't do that yet. So when you make viruses, though, the quasi-species that you produce are the ones that have gone through the selection in you, and when they reinfect someone, then the selection imposed by transmission through the air or on a surface going into the new person, all of that puts pressure on the population and changes it. There may be some viruses that don't last very long floating in the air, so they're selected against, and the others do. They come infect you. So basically, when you have small populations of particles replicating, you have extreme fluctuations in both the genotype and the phenotype. So virus spreading from one person to another can change extensively as a result of the quasi-species. So it's very important that you get this. And so to illustrate this further, this is a sequence I just took out of GenBank. You can go to PubMed and search for any virus and pull up a sequence like this. Does anybody know what virus this is? It's polio. It's getting good. Yeah, I'm impressed. So that's polio, the first 1,000 bases. And it's a defined sequence. But polio virus is not a defined sequence. This is the average that you get when you sequence a population of viruses. So when you think about it, how is this sequence determined? Well, one way that you can do it is you can extract viral RNA from a particle and convert it to DNA using reverse transcriptase, and then sequence it. So you're really only sequencing a very small fraction, maybe one of the RNAs in the population. And that's what we have here. You can do the same thing today by PCR. In fact, you see all these influenza sequences in GenBank. They're all produced in the same way. They have no relationship to the population that's inside of you. So when you look at this, just remember, whenever you look at a virus sequence, remember it's of some particle. Maybe there's no particle in the preparation that even looks like this. Maybe it's a rare particle. Who knows? Yes?
So the term quasi-species and the concept of quasi-species isn't essentially different from that of species. It's just a more extreme version. The species themselves are not defined by consensus sequences.
What defines a species?
Well, it's very clear. It's a big philosophical problem. But it's agreed upon that there aren't, at least most people agree that there aren't sort of necessary and sufficient conditions for defining a...
Yeah, well, the order of difference is several magnitudes, so it's huge. So and it could be a totally different phenomenon in that sense because of the magnitude. But yes, at its simplest level, it's a numbers issue, right? So I don't know what the sequences mean in terms of what we study in the laboratory. You can change some of these sequences and have effects on viral populations, but who knows what it's doing to the actual quasi-species. So we so far haven't been able to study this. At some point we will, and perhaps we'll understand even more about the relationship of the genome to the property of a virus. So for example, you have influenza viruses in Mexico causing lethal disease, and the same virus, you sequence it, and it's the same virus in New York that's not causing lethal disease. And people puzzle, how can it be that's the same sequence? Well, maybe the quasi-species is different in Mexico than in New York. I'm not saying it is, because no one knows, but it's certainly a possibility that if we could dig down all the way down to the quasi-species level, we might find differences. So for any virus population, the genome cluster around a consensus or average, and that's what you see in PubMed, but every genome can be different from every other genome. And it's very unlikely that a genome with this sequence is actually replicating in the population. So when the US government paid to have a human genome sequence years ago, that was a good representation of what's in many of us with differences. But when you sequence a virus, it certainly is in the same sequence space, but the details are all very different. So I want you to be skeptical about virus sequences, not that they're correct or not, but that they're an average which doesn't really represent the population. So when we have a quasi-species, we can have survival of the fittest. If the virus goes to a new host, a rare genome with a particular mutation could survive selection. So if it goes from just from me to one of you is a selection process, because we're all different in some way. And that can select for a certain mutation in the viral genome. So you have particular mutations which are selected for and allow the virus to grow better in the next host, say. But there are also other linked but unselected mutations. So the mutations on the same RNA, which don't confer any advantage or disadvantage, they're coming along for the ride. They're getting a free ride. So after selection, you get a diverse population, and the only things in common are the mutations that are selected. You have lots of other changes that came along. They're neutral, if you will, and that's what maintains the quasi-species in addition to error-prone replication. So the idea that we have a hugely variant population that changes at every step, every time it replicates, goes to a new cell during transmission, it's being selected on, that's the important point here. That's what I want you to take away. Now, DNA viruses are different. They have different lifestyles. They tend to have a narrow host range, and I'm glad Dr. Silverstein isn't here today to correct me, because he would defend his topics of study, of course. But in general, compared to RNA viruses, DNA viruses have a narrower host range. They tend to produce persistent infections, which you may remember are characterized by lower total virion production over time. So there's not as much progeny, not as much opportunity for variation, but so not much replication. But the most important point is that the genome replication is not as error-prone as that of RNA viruses. The DNA polymerases make mistakes, as do RNA polymerases, but there's a correction mechanism, as we talked about last time. So there's proofreading available for DNA viruses. So because they have less replication and they have proofreading, they generate less diversity and they evolve more slowly than RNA viruses. Okay, so that is the quasi-species. That's the diversity that is part of evolution. Let's talk about selection. Selection is what are the pressures that are applied to genomes that result in the enrichment of particular quasi-species, if you will. So selection, and this is an interesting statement, selection favors both the creation of diversity as well as single dominant mutations. So that sounds counterintuitive, right? How can you be diverse and yet have single dominant mutations? Well, that's because there have to be certain mutations in the population. Remember, it's a population thing that are selected for to survive in a certain host or under certain conditions. But you have to maintain the population as a diverse one. If you don't, you will suffer, as you'll see in a few moments. So selection pulls out the mutations that are important for that particular event. And remember, at the same time, all the unselected mutations go along for the ride, and those are important as well, because who knows, someday they may be important in another environment. So this whole idea of diversity as a consequence of increased mutation rate is an interesting one, and you may wonder, well, why do we make mistakes? Is it necessary to make mistakes? And in fact, it is. And we can show that experimentally. So increased mutation rates are selected for during virus evolution. Mutation is actually good, up to a certain point, of course. Mutation is good for viral populations. You can make mutant or altered viral polymerases that reduce the frequency of misincorporation errors. These were first done in E. coli. You can actually, even though E. coli is a DNA-based organism, it does make mistakes. You can isolate mutant E. coli. They're called anti-mutators. They have changes in the DNA polymerase, so they make fewer errors. You can do the same with viruses. There is a mutation in the poliovirus polymerase, a single amino acid change that makes the polymerase, the RNA polymerase, less error-prone. And so it makes about 3 times fewer errors than does the wild-type polymerase. Yes?
Can you do the opposite and make it more error-prone?
Yes, you can do that. And you have other issues with that as well. Sure, you can go the other way. But you know, in nature, the polymerases have evolved to be balanced, not too few errors and not too many. So when you study these mutants, what you find, either bacteria or viruses, they don't have a selective advantage. So if you take a wild type and a mutant, where the mutant makes fewer errors and you mix them, the wild type always outcompetes the mutant. So making fewer errors doesn't help you in competition. And that's for bacteria and viruses. Lower rates aren't selected for. So in a mixed experiment, the viruses or the bacteria with the higher mutation rates are selected for in the end, not the ones with lower. And mutants that are like this, that make fewer errors, are often less pathogenic in animal models. This has been shown, for example, with polio. That mutant in the polio polymerase that makes fewer errors in a mouse is unable to cause significant disease. So the bottom line, or the conclusion, is that mutation is part of what is selected for during evolution. It's essential. It's good for virus populations. It's good for us as well. If we didn't mutate, we wouldn't be able to adapt to our environment. Of course, along with mutations, you have negative consequences as well. You have various diseases, but you have to have a balance, obviously, between the two. So the conclusion is that high mutation rates are a positive force in virus evolution. But you can't make too many mutations, right? Obviously, you have to go to a certain point, and that point is called the error threshold, where beyond which you don't want to venture, because then you will die. So we like to make a lot of mutations. That's what we've said so far. That's our conclusions. But there must be a point where selection and survival balance fidelity and mutation rate. So as I was just saying, you want variability, but not so much that you make diseased organisms. And so that point is the error threshold. If you exceed it, you die, or if you're a virus, you can't replicate anymore. And if you live too far below the threshold, you can't make enough mutations to survive selection. So the anti-mutator mutants that we just discussed live way below the error threshold, and they can't survive. They just cannot compete in nature. Now, RNA viruses, interestingly, live very close, or exist very close to their error threshold. You can push them a little bit higher in mutation frequency, and they will extinguish themselves because they exceed the error threshold. We'll see that in a minute. Whereas DNA viruses are way below the error threshold. You can make DNA viruses mutate more, and they will be okay up to a certain point. So there's a lot more leeway between where DNA viruses are in the error threshold compared to RNA viruses. Okay. So that's an important concept, this error threshold. So let's do an experiment that illustrates it. We have this drug called 5-azacitidine, which is a mutator. It induces mutations in DNA. And we infect a cell culture with a DNA virus, and then we put this drug in the cell culture. So the 5-azac Every time the polymerase has a G, it wants to put in a C. It puts in instead 5-aza C because you've got a lot of it in the medium. But when that strand is then copied again, it templates as a T. So it doesn't, the polymerase doesn't see it as a C. So normally if it were a C, the polymerase would put a G in. But aza C is seen as a T, so the polymerase puts an A in. So you have a mutation. So this induces mutations in the DNA. All right? And so what happens to the DNA virus? The mutation rate increases several orders of magnitude. So you can take the yield of this infection with DNA viruses, and you can measure mutation rates. You could just sequence the genomes, or you could look at a variety of other parameters to measure mutation rate. The DNA virus mutation rate goes up 10, 100, maybe 1,000-fold. You do the same experiment with an RNA virus, you get a 2 or 3-fold increase in mutation frequency. You don't get any more than that. So the DNA viruses goes up 100-fold in mutation rate. With the 5-aza C, the RNA virus 2 to 3-fold. Everybody understand that? It's an important concept. So this shows you that the RNA viruses are already almost at the maximum of their error frequency. They can't go much beyond it. You can't push them. We say that RNA viruses exist at or near their error threshold. Now, this is an interesting property because a number of people have suggested that maybe we could make antivirals that push RNA viruses over the error threshold, mutate them to death, in other words. And some of those are actually being tested. So it's an interesting concept. So you don't actually inhibit any particular part of the virus life cycle. You don't inhibit an enzyme or a neuraminidase or something else. You just have the virus mutate itself to death. So it has mutations in many proteins and therefore it doesn't replicate in you. Why does that work? Because we have DNA-based genomes and we can accommodate the increased error rate when treated with these drugs. Okay, so that's error threshold and that's the evidence for it. Now, in connection with quasi-species and error rates, there's a concept called the genetic bottleneck that we need to talk about. It's exactly as it sounds. And if you've taken a genetics course, you've heard about this in other terms. It turns out that when you have a small population and you put a lot of pressure on that population, you get loss of diversity. You get a lot of mutations in the genome, loss of diversity. This is called the genetic bottleneck. So for example, let's do a bottleneck experiment with an RNA virus. We do a plaque assay and we pick a plaque. And then we take that plaque and reinfect fresh cells with it and pick another plaque. And we keep doing that on and on. We do it over and over. And so here's the process illustrated. You do a plaque assay, you pick a plaque, you put it in a little buffer, and you do another plaque assay on that, and then you get plaques, and you keep picking plaques. You do this over and over again. This is creating a bottleneck. So what happens when you do this? You get a bottleneck because what you're doing is restricting all the viruses that you're reinfecting cells with to just the viruses found in a single plaque, which isn't that many. It may be a few thousand at the most. All right, so one virus gives rise to a few thousand. You then take those and use them to infect a new cell. So you're restricting diversity in your reinfection. So here's another way of looking at it. You have a bottle of viruses, of blue and yellow viruses. Of course, they should each be a different color, right, because it's a quasi-species. When you pour a little bit into a culture dish, only a few come out because there's a bottleneck. And then when you go and grow those up in cells, you have a different population than what you started with, because what came out is just a fraction of what was in the original bottle. So that's why it's called a bottleneck. This illustration comes from a bottleneck in a eukaryotic organism, which is why there are only 2 alleles, if you will. But again, if it were a virus, it would be all different colored balls, and there would just be a few different colors coming through the bottleneck, and those would give rise to a very different population. So in this case, the bottleneck that we're applying is picking a single plaque and using just a very few viruses to infect new cells, as opposed to growing viruses in a few million cells on a plate and taking that total population. So we're taking just a few that are in a plaque. Okay, so after you do this 20 to 30 times, you get very sick populations. They don't grow well. And they're much less fit than the original population. And it's interesting because the environment's constant, right? You're just going from plaque to plaque. You have the same cells, the same incubator in your lab, same temperature, the same medium. So there's no apparent selection except making a plaque. Yet the viruses eventually get lower and lower and tighter, and you can barely grow them. And we have done this in the lab with our viruses. It absolutely works beautifully. I had one student a few years ago who was doing an experiment, and after 30 plaque passages, she said, "My virus isn't growing anymore." And I said, "Did you take my virology course last year?" And they said, "Ah, yes, it must be a bottleneck." Yes, that's what it was. But why does this happen then? Why do you get sick viruses produced by doing this bottlenecking? Well, the answer is something called Muller's ratchet, which was devised years ago for other genetic systems. And he said that small asexual populations decline in fitness over time if the mutation rate is high. So viruses are asexual, and they have a high mutation rate. So this applies to viruses as well. So normally, remember, RNA viruses make a lot of mutations. They're at their error threshold. By restricting the population to the viruses in a plaque, it's just a subset of the whole quasi-species that's possible. Eventually, those viruses, in order to survive, they accumulate more and more mutations in their genome, and they exceed the error threshold. And they become sick because they're going over the error threshold. So why is it a ratchet? So that's a ratchet, in case you don't know what the metaphor means. And a ratchet works, you can turn the wheel and push the ratchet forward, and there's this little lock here that prevents it from going back. So that's why there's this metaphor. Each mutation works like a ratchet, allowing the gear to move forward but not backward. And every time the virus replicates, you have a ratchet clicking as mutations accumulate at every cycle, and you can't get rid of them. So eventually, the fitness plummets. So here are a couple of examples of fitness decline after passage through a bottleneck. These are different viruses, number of bottleneck passages, and the percent decrease in fitness after these number of passages. So you can see this really works. Now here's an interesting question. HIV doesn't form plaques in any kind of cell. So how could you do this experiment that I've just told you? You could answer it now, or I could put it on the final. So remember, taking a plaque, you have just a few viruses, right? That's the key. You don't have a big population of viruses. You have a few thousand viruses in a plaque. How could you duplicate it for a virus that doesn't plaque? It's really simple.
Just take a couple steps or in the tissue.
Just make a dilution. Use a small amount of virus. Each time you dilute it and use a small amount of virus to pass. It would basically be the same thing. So that's what they do for HIV. And it's not as effective. Well, it is effective. I'm sorry. It basically does the same thing. And for any virus that doesn't form plaques, it's sort of like doing a limit dilution and just picking a few virions and passing them. Yeah?
Isn't it effective because in the body it's incorporated in the cell? So you never really...
So are you asking whether you get bottleneck effects in the real world? Okay. I think that's 2 slides down. So how do you avoid the ratchet? Subject a more diverse population to passage. Don't just pick a plaque. Maybe if you mixed some plaques together, you would avoid it. In fact, that would be the case. If you mix 5 or 10 plaques together and do this passage, you don't get a bottleneck. So you get more diversity, and you have the ability to contribute to the quasi-species. And that allows you to produce genomes that are fit each time that they replicate. And so you have lots of diverse genomes in this population. They can recombine or reassort or just generate diversity that wouldn't be possible when you're just using the limited numbers of virus in a single plaque. So you pick a couple of plaques and you do the experiment with them. When you do that, you don't get a bottleneck. Okay? So when you're doing this mixed plaque passage, a recombinant may arise that's very rare, but it might predominate during the passage and grow out and be the dominant population. And you're allowing that to happen because you start with a more diverse population of virus. And you prevent that from happening by restricting the bottleneck to a small passage, to a small inoculum. So the message is very simple. Diversity is important for the survival of individual members. So again, this is why virology is a population study. Each individual virus will not survive unless the population is diverse. You remove the diversity, which is what you do by bottlenecking, and the virus suffers. So here we go to your question, bottlenecks in the real world. This is found in nature quite often. So you get infected by influenza virus from someone sneezing. There's not a lot of virus in those droplets that go in you, probably as much as in a plaque. So that's subjecting the virus to a bottleneck. Of course, once it's in you, it then has the ability to replicate and amplify in many, many cells. So the effects of the bottleneck aren't seen. A latent virus activated from a limited population of cells, if those viruses don't reinfect new cells, that's going to be a bottleneck right there. Or when an insect bites you and delivers viruses, there's not a lot of virus in there, it's a bottleneck. And if that virus, say, replicated in you and then another insect took it and moved it to another person, if it did that a number of times, it would constitute a real bottleneck. But in practice, that doesn't happen in nature. So we don't see the effects of bottlenecks. But let's say that there are situations where bottlenecks occur. How would natural infections escape the ratchet? One way is by recombination or reassortment. So if you have created by the bottleneck genomes with excess mutations, which are shown by the lightning bolts here, you can simply have recombination occur during replication and you make a healthy viral recombinant. So if you have enough population, this is what can happen and you avoid the ratchet. So that's exchange of genetic information among viruses, and it happens within populations all the time in order to escape bottlenecks and also to provide additional diversity in addition to the mutation that occurs at the level of RNA synthesis. But there is other types of exchange of genetic information that occurs. So one example is to avoid the ratchet, which we've talked about. And this happens in cells infected with RNA viruses with segmented genomes, like influenza viruses, which we have talked about, reoviruses. Reassortment provides additional diversity. And we'll talk about how it provides variation in influenza viruses. So we now can look at a full picture of the quasi-species and explain some of the population effects. So here is the quasi-species that we looked at originally. If you take this whole population and you infect a new cell or a new host, mutations are going to be selected out of this population that improve replication in that host under those conditions. So the viruses that come out of this will have increased fitness. So my quasi-species going to another person, certain mutations will be selected for and increase fitness. And this is what happens when we do large population passages. If we do a bottleneck, on the other hand, we squeeze this population through a bottleneck by picking a plaque and passaging it. Basically, we're taking one or a few genomes and squeezing them through the bottleneck, and then in a new host they expand. But now you see that all of these genomes have the original mutations that went through the bottleneck. They have others as well, but if you keep doing this over and over again and selecting, you will have the same constellation of mutations. There will be still increased error rate, and you push the virus over the error threshold, and you have decreased fitness, which is what we saw in the plaque-to-plaque passage. So this is why another reason why population diversity is important. It allows viruses to survive under different conditions. So let's talk a little bit about some other kinds of selection that occur. And this is specifically genetic drift and genetic shift. And we've talked about these a little in terms of influenza viruses, but they happen in other viruses as well. And they're a consequence of applying selection pressure to a quasi-species. So, for example, you make an immune response to viruses when they infect you. The antibodies and the T cells exert selection pressure on the viruses. Now, if the virus were all one genotype, it would be eliminated. But it's a quasi-species, and in that quasi-species are antigenic variants that can escape either the antibodies or the cytotoxic T cells. So your immune response applies selection, and the virus population changes accordingly. So when that happens, we have the two kinds of change. We have antigenic drift, or genetic drift, depending on what level we're talking about. And this happens with immune selection. And then we have shift, which usually is a consequence of recombination or reassortment. So drift are the point mutations that occur from error-prone replication that are selected for by immune systems. Shift is the change caused by recombination or reassortment. So let's talk about that in the context of influenza viruses, which by now you know really well. And you know they have surface glycoproteins, the HA and the NA, and that's how we classify these viruses. So there are 15 different hemagglutinins in the world that we know of, and 9 different neuraminases. So there are different combinations of H and N, H1N1, H2N2, etc. All 15 of these can infect birds, and the main human viruses are H1, H2, and H3. H5 periodically goes from birds to people, but so far it hasn't transmitted among people. So this is how reassortment gives rise to diversity for influenza virus. So here, let's start in 1918. You have introduction of an avian virus into humans. And nobody's immune to this avian influenza virus, so it spreads globally, causes influenza, and in fact, it circulates in people until 1957. And in 1957, another virus emerged which gets other genes, including the HA and the NA genes, from a different kind of bird influenza virus, an H2N2. So this virus emerging in 1957 is an H2N2. There's no immunity in the population. So this is selection. The original virus, now the H1N1, there's a lot of population immunity, and now a new virus emerges accidentally from a bird population, which is recombined with the H1N1. There's no immunity to it, so this predominates. This virus replicates like crazy, causes a pandemic, and spreads globally. So that's how selection, and in particular immune selection, is selecting for viral reassortants with different antigenic properties. And this goes on every so many years. In 1968, another shift to an H3N2, and again from genes donated from a bird. And this is what happened last year to give rise to the latest pandemic strain, the 2009 H1N1, a series of reassortants happening as far back as 1990 among swine viruses, human viruses, and avian viruses. So there are separate reassortment events that occurred over a long period of time, so that this virus has the H1 and the N1, which is highly related to the 1918 H1N1, and then other segments from different viruses. Again, it's a result of immune selection. The virus that's introduced accidentally into the population from pigs or birds isn't recognized by human serum, so it has a selective advantage. And these viruses wouldn't emerge if there weren't mechanisms of diversity, in this case reassortment. But every year viruses emerge, slightly different influenza viruses, by drift, which is a consequence of error-prone replication. So here's an example of that. Remember, influenza virus, enveloped virus with hemagglutinin and neuraminidase glycoproteins. This is the three-dimensional structure of the hemagglutinin, which is this protein here. Remember, this protein attaches to the cell receptor. The receptor binding site is up here at the top of the molecule. Over years, you have mutations at these colored sites at the very tip of the HA. So these are actually changes in the H1 hemagglutinin from 1918 to the present. And you can see they're all up here. This happens to be where the antibodies bind that will neutralize this virus. So the virus makes errors during its replication. Those are incorporated into the hemagglutinin. And as antibodies remove the hemagglutinins that haven't changed, what's left? The ones that are slightly different, and they replicate, and they are selected for in the population. So again, it all goes back to error-prone replication in terms of antigenic drift. Okay? Now, exchange of genetic information happens among viruses, but it also happens between viruses and hosts, and that's another way that viruses evolve. So sometimes you can get cellular genetic information inserted into a viral genome that changes their properties. So one example is the production of a virulent virus. It's called bovine viral diarrhea virus. Okay? So this is a virus. We'll talk about that in a moment. This is a virus that infects cows, of course. Retroviruses, remember, pick up oncogenes from the cell. It's an accident, but they do pick it up and they become oncogenic as a consequence. And many viruses have picked up immune system genes from the cell. They've changed slightly, and now they're antagonists of the immune system. So those are three examples of how viruses pick up genes from the host cell by recombination, and it modifies their properties. So let's look at this bovine viral diarrhea virus. It's very interesting. So many cows are infected with this virus. They're infected in utero, and the fetal cows are fine. They get a persistent infection by this virus here on the top. This is the genome of that virus. It's called non-cytopathic bovine viral diarrhea virus. It's non-cytopathic. It doesn't kill cells. It doesn't cause disease in the cow as long as it's in utero. When these cows are born, so the virus is replicating during gestation. When the cows are born, the virus kills them. It becomes virulent, and it becomes cytopathic down here. And it does so because during the time it was replicating in utero, in the fetal cow, it picked up a piece of DNA from the host. And that made the virus more virulent. So the DNA it picks up is actually a piece of DNA that introduces a protease cleavage site right here in front of this protein NS3. So it picks up a site for cleavage by a cellular protease, and for some reason which we don't understand, this makes the virus more virulent. And these viruses outgrow the non-cytopathic viruses. That's why they are selected for. They also happen to cause more disease. So keep that in mind, this association between replicating better and disease, because it's an interesting question that we'll get back to. Another example, yes?
So can that kind of recombination only happen with RNA viruses because they either have to be retroviruses or picking up nucleic acids that aren't in
No, it could happen with DNA viruses because if the DNA virus is replicating in the nucleus, the polymerase could switch back and forth between nuclear DNA and viral DNA. So it can happen in either case. And there are examples of that too. Here's an example of this kind of recombination that has led to the production of a new virus. This is theoretical, but very interesting. You remember circoviruses? These are viruses with single-stranded DNA genomes. They're vertebrate viruses. Well, there's an analog in plants. They're called nanoviruses. They have the same DNA structure. These viruses have a few genes only, and one of the genes is called the rep gene. It encodes a protein important for replication. That protein is very similar between the circo and the nanoviruses. In fact, it looks like a hybrid of a circovirus and a nanovirus protein. So this implies that these viruses came together at some point. In addition, there are bits of the rep protein that have homology with a third virus, the calicivirus, which is an RNA virus that infects mammals. So the idea arose by sequence analysis of these genomes that first there was nanovirus, a plant virus. At some point, some vertebrate ate a plant or drank sap with nanovirus in it, and within the nanovirus population, there was a virus, the quasi-species, one of the quasi-species could grow in a vertebrate. It took off, and that became circoviruses. And then in the mammal or the vertebrate, it then recombined with part of the calicivirus genome, this RNA binding protein, and that gives us our present-day circoviruses and nanoviruses. Another idea of how recombination can give rise to diversity. So here's an interesting question which I think we've touched on very briefly in this course, and there's no answer to it, but it's interesting to talk about just for a few minutes. Is virulence a positive or negative selection trait? Do viruses want to get more virulent? So the idea is that if a virus is more virulent, it kills the host quicker and would reduce its transmissibility because the virus can't spread to another host as readily if the host is dead. Okay? So the idea is that maybe viruses evolved to be maximally infectious and avirulent. If this theory is true, this would be the observation. But it's not true, unfortunately. We don't see this. We have many infections that kill people. We have them that kill very quickly. We have some that go on for years and then kill the host. We have viruses whose virulence differ in one species versus another, even though they're equally transmissible. So there's no, it looks like there is no reason to assume that this is correct. Now, I've always felt that transmissibility is what is selected for, but in some diseases, being more virulent goes along with transmissibility. So this number R0 is simply a measure of the number of hosts that are infected by a single infected host. And for some diseases, if the virus becomes more virulent, this transmissibility also increases. So you can imagine if a virus makes you cough more, that's a more virulent virus, but it also improves transmissibility. The bovine diarrhea virus that we just talked about, the main selection for that virus is increased replication rate. It replicates faster because it picked up this cellular gene. It also happens to kill the cow, but that's really a side effect. And remember, this virus has been replicating persistently in utero for many, many months. So it's had ample opportunity to spread. So maybe under certain conditions, increased virulence can be tolerated as long as transmissibility is maintained. So what I want to leave you with, think about this, whether virulence is a positive or negative selection. I don't think there's a single answer. I think it depends on the specific virus and the host and the kind of disease that is formed. And if you're interested in this issue, there's a very interesting book by Paul Ewald, Evolution of Infectious Disease. And the first paragraph of this book is completely relevant to our discussion. Few ideas have been so ingrained in the literature of medicine and parasitology as the idea that parasites should evolve toward benign coexistence with their hosts. Few ideas in science have been so widely accepted with so little evidence. Well, he's a little opinionated. There is a lot of evidence for this. And few ideas are at so odds with the fundamental principles on which they are based with such great potential for missed opportunity. So his theory is that it depends on the virus and the host, whether transmissibility and virulence are positive selection factors or not. It's worth reading. Okay, so let's do some experiments in virus evolution in nature. Here's an interesting one, which was done in Australia. So in 1859, they brought this European rabbit there because they wanted to hunt it. So the hunters released these rabbits, they multiplied, and they would go out and shoot them for game, sport. But there are no predators of this rabbit, and so they multiplied like crazy, and they were all over the place, and they said, we have a problem here, let's get rid of these rabbits. There weren't enough hunters to kill them all, so what they thought was to kill them with a virus. They're going to release a virus. So they used this a pox virus. It's called Leporipox virus. It was released in the 1950s on the continent of Australia. And the natural host of this virus is the cottontail rabbit, brush rabbit, and a rabbit of Central and South America. It's spread by mosquitoes in warm months, and these rabbits just get warts. It doesn't kill them, but the European rabbits are different, so they're killed by this virus. 99% fatal. So they thought in Australia, the Australian virologists thought, let's release this virus and it'll kill all the rabbits. Okay, so what happened? They released this virus the first year. It killed 99.8% of the rabbits. Okay, well, it killed 8.8% of the ones that it infected, but it didn't infect everyone. But the second year, mortality rate was only 25%. And subsequent years, it was even lower than the reproductive rate of the rabbits, so it didn't work. So now we still have European rabbits running around Australia, and this failed. So why was that? As you know, both rabbits and viruses multiply prodigiously. Okay, and that's the key here. So the virus evolves, which you understand by now because that's easy to figure out. Kill fewer rabbits. Extend their life. It turns out that this virus doesn't spread well in the winter. So the virus evolved so that it would infect rabbits in the spring and then stay with them over the winter until the next spring. So it would persistently infect them so that it could reinfect new rabbits in the spring. Why? Because it selected for, among the quasi-species, viruses that are able to do that. That's why quasi-species is so great. So it evolved, the virus evolved, but also the rabbits became more resistant because they can make many generations. You could select for rabbits that could survive virus infection. So they became tolerant of the virus. And this is what Paul Ewald doesn't like, as predicted for an evolving host coming to an equilibrium. So I'm saying the virus and the rabbit came to equilibrium. And Paul Ewald will say there's no evidence for that, but this is pretty good evidence. And there are other stories as well. So what did we learn from this? You always get what you select, but you often don't get what you want. It's a paraphrase of some song somewhere. So that one you recognize, right? So eliminating rabbits with a virus was a bad idea because there are selective forces that you can't control. The virus and the rabbits are changing. You have no control over that. If the virus were one genotype that never changed, you might be able to do this, but it's not the case. In Australia, though, they apparently learned nothing because now they're doing experiments to control rabbits using a different virus. So they're using this Khaleesi virus. A Khaleesi virus. So I read about this. You can find this online. They did some experiments in one part of the island, restricted, closed off to see if this would work, and the virus escaped. And it's now infecting the rabbit population, and they can't control it, and people are afraid that their pets are going to get infected as well. So that's another disaster. The moral is don't use viruses to control wild animals. But that's a nice experiment in evolution. So in the last, yes?
You're saying use 2 or 3 viruses at the same time. It seems like it meant
2 or 3 different viruses?
Yeah. So like
What kind of virus would have a better chance? Well, actually, this was a DNA virus.
virus and the first one was
Yeah, the first one was a pox, so that was a DNA.
at the same time. Then the one would have eliminated 98
I don't know. Do you want to go there and try it out? There's still unknowns. That's the problem. You don't know how they're going to interact. Maybe they'll reassort or recombine and make a new, more lethal virus. Who knows?
Right. It's similar to the way we control HIV infections, right?
Different drugs?
We reduce the chance that that there will be a mutation which has multiple resistance.
I don't see why the viruses can't change and be selected independently in your experiment as well.
Well, because if one of the viruses is so effective as to wipe out 98% of the hosts,
Mm-hmm.
then there are no hosts left for the virus to evolve or to mutate.
Yeah. So, but the important thing is you have to infect. You have to have efficient infection with all 3 at the same time, which may not happen in nature, right? It's an interesting idea. So, a little bit of time left. Let's talk about the origin of viruses. We don't know where viruses came from because we don't have a fossil record, but there are theories based on nucleotide sequence. There's this regressive theories that says they came from parasites that lost genes. The cellular origin theory, they came from cell components, maybe a mitochondria or a nucleus that gained the ability to replicate, or maybe they co-evolved. I think this is a good idea. I don't see why viruses couldn't have been around even before cells, just nucleic acids replicating in organic soups, primordial soups. But as I said, there are no fossil records, and we don't have very old viruses either. Wouldn't it be great to go back in time millions of years and take samples and sequence them? I think this would be the best use of a time machine. Anyway, you could learn so much. Hey, so it's hard to test hypotheses, so we study contemporary viruses and we try and make guesses based on phylogeny. So I said we don't have a fossil record. Is there an exception to that, though? Can you think of one? Not fossils in the sense of, rocks, but something else. Yeah, that's right. So retroviruses in genomes are sort of fossils, and you can trace them back phylogenetically very effectively. You can predict when they entered the genome, and by looking at the relationship in different species, and if you know how those have evolved, you can get some very nice ideas. So some interesting facts on the origin of viruses. There are viruses in all 3 domains of life. They have homologous features, which suggest that there might have been a precursor to all of those viruses. The oldest ones may be the viruses of green algae. Their genomes are at the root That is, the left side of all eukaryotic sequences. If you make a tree from left to right, the DNA viruses of green algae are at the left, but there may be more that we haven't discovered yet. And as we sequence more and more genomes, we find genes with no obvious homology. So there's a lot of information still to be learned from this approach. We have these very large viral genomes like the Mimi viruses, the 1.2 million base pairs, these phycodNA viruses or the blue-green algae viruses. They are very similar. They have sequence coherence. They're not mixtures of different viruses. And so this suggests that they had a common origin. But within the family, there's no homology among the families. And so it's hard to say that they arose by acquiring genes from an old precursor. So we don't actually know how these very large viral genomes evolved. Someone recently has suggested that because the Mimi viruses replicate in amoebae are phagocytic. They take up a lot of junk from the environment, bacteria and other organisms. And maybe the viruses incorporate the DNA from those organisms in them. We do a lot of metagenomic analysis to study evolution. We know a lot of viral sequences right now. We can do microarrays to sample the environment for genomes. We can sequence viruses from water, and that's all been done. And when we do this, we find all new sequences all the time in the oceans and in the water. Now, you can get an idea on the origins by doing phylogenetic analyses of all the viruses that you have sequenced, and by calculating the mutation rate, you can backdate the viruses. When we do that with herpes viruses, it's been estimated that they arose between 180 and 220 million years ago. So that's not too long ago on the evolutionary scale of the Earth. Here is about 250 million years ago. So that would mean that the dinosaurs had herpes. There are also oyster and amphibian herpes viruses in fish, which are around today. They are herpes viruses, but the sequences with the other herpes viruses is very low. So this may have been a very early branch of the family. So as we sequence more and more, we can do studies like this. Here are two examples of how we can figure out the origins of certain viruses. Smallpox, the sequence analysis of a number of isolates show that they cluster geographically. And the gene constant among all these viruses, the gene content is constant, very little diversity. So that means the viruses recently went into people from some animal. And the only member with homology to smallpox is a gerbil pox virus. So all the other viruses are not as similar. So the idea is that many years ago, there was a zoonotic infection of a human from an infected gerbil, and that gave rise to smallpox. This would have to be a long time ago, obviously. Papillomaviruses and polyomaviruses, these small DNA viruses, their origins are very interesting. They seem to have co-evolved with the host because you can look at the sequences of these genomes and map them with racial and geographic distributions of human populations. So you can associate specific genotypes of these viruses with the migrations of populations over time. So JC virus, for example, provides a marker for human migration from Asia to the Americas. It's quite remarkable. And why is that? How could a virus mark a human migration? Well, as you know, most viruses are spread horizontally by contamination or by respiratory secretions. But these viruses are spread from mother to child. So it's almost like a vertical transmission. So that would match population migration. Measles virus seems to have come from a cow pathogen called rinderpest. It's closely related by sequence analysis. So when humans first began to domesticate cattle many years ago, we probably acquired the infection from them. Before that time, we weren't very close to the animals, so there was no opportunity for the virus to infect us. And we think that the infection was established about 5,000 years ago in the Middle East when populations began to congregate and domesticated cattle. And this virus, after being established in humans, spread around the world by colonization. It didn't reach the Americas until the 16th century. It was brought there by colonizers, and of course it decimated native populations. So those are some of the detective work that we can do to track the origin of viruses. But obviously it doesn't go very far back, and it would be interesting to do that. So the fundamental properties both constrain and drive virus evolution. We can, despite all the mutation that I've told you about today, we can still recognize a herpes virus or an influenza virus by its sequence analysis. So there's some constraint to maintain a virus within its sequence space. How is that done? The viruses obviously have a number of characteristics that they have to maintain. And they have to function within those constraints. And what are they? Anything that goes beyond the sequence space of a virus doesn't survive selection. If a herpes virus changed too much, it wouldn't survive. So the genome is one constant. DNA can't become RNA, or vice versa. You can't change your replication strategy. Remember, you have to interact with a variety of host proteins, so you can't change that. The nature of the capsid, you can't go from an icosahedral capsid to an envelope virus. You have to stay within your sequence space. And during the host infection, there's also selection. You can't readily change hosts, although that's probably the most plastic of all the genome constraints. So here's a great example of selection for survival. Yes, do you have a question?
Yes. DNA cannot become RNA or vice versa. I guess it's just retrovirus.
Well, so what I mean by that is that a virus that has a DNA genome can't mutate so that it now has an RNA genome, and that perpetuates. It's true that retroviruses copy their RNA into DNA at one point, but then it goes back to RNA, and that's packaged in the virion. Right? HIV, at the end of AIDS, you're making billions and billions of virus particles. These viruses are called T-cell tropic viruses. They like to use the CXCR4 co-receptor. But when this virus is spread to someone else and infects them, the first genomes that replicate in the recipient are viruses that prefer the CCR5 receptor. So you make tons of CXCR4 viruses at the end of infection. When you infect someone else, the viruses that succeed in them are the R5 viruses. So the selection is extreme for co-receptor usage. So this is a bottleneck. This is an example of a bottleneck. Only if you pass through this, the bottleneck is transmission from one person to another. And apparently viruses with CCR5 preference are the ones that go through that bottleneck. So the ones that kill you, the viruses that devastate your immune system, are not the most fit for infection of new hosts. You've evolved a population within you over a 10-year period that's excellent at infecting your immune system, but not as good as passing to another person. And if you didn't have diversity in these genomes, you wouldn't be able to be transmitted. So new viruses will only arise from existing ones. They will not arise de novo. They will evolve from what we have. So how will that happen? What's the number of all possible mutations of a viral genome? It's so large that we can't test all the possibilities. And sequence comparisons have shown that over half of the genome of most RNA viruses can accommodate mutations. So if you have a 10 kilobase viral genome, there are 4 to the 5,000 sequence permutations that define all possible mutants. 4 to the 5,000. Is that a big number? Well, it's huge because there are 4 to the 135 atoms in the visible universe. So this is just the amount that one virus with a 10 kb genome could change. So it's phenomenal. It's a phenomenal number. And this is what I'm telling you from the start, that viral variation is almost impossible to believe. So, and I'll leave you with HIV again. Remember, it undergoes incredible replication during the 10-year period of AIDS with huge opportunities for selected. There are millions of people infected by the virus. We produce billions of genomes a day. So we have 10 to the 6 genomes each day on Earth of this virus. And the mutation rates are such that there are mutations in every genome that is produced, and we have mutants resistant to every combination of drug that we're using now or that we will ever develop. And I'll leave you with the conclusion that evolution is relentless. We'll always have new viral mutants arising, and future viruses will come for what we have now. There'll be mutants, recombinants, or reassortants. And think that we only know about 1% of the viruses that exist right now. So you can imagine what potential for variability still lies. So it took 8 million years for simian primates to change 2% of their genomes to become humans. 8 million years. Polio can change 2% of its DNA structure, or its RNA structure, in 5 days. So can you imagine, that's about the time it takes for the virus to get from the mouth to the gut. It's just 5 days, 2% of its genome has changed. Can you imagine what a virus would do with 8 million years?